audio. We're very lucky to have Marco Salemi speaking to us today. Marco is well known for his work in molecular epidemiology, intra-host virus evolution, and the application of phylogenetic and population genetic methods to the study of human and simian pathogenic viruses. He was a Marie Curie Fellow at the Reagan Institute and the Catholic University of Leuven for his PhD, did postdoctoral work with Walter Fitch at UC Irvine. He's currently at the University of Florida. So we're going to get started, and you can see that today we have our very first live audience. It's not just me, and I'm very excited about that. So we'll hear Marco today talking about the phylogenetic challenges in the Retroviridae branch of the tree of life. Take it away, Marco.
Okay, thank you. Can you guys hear me? Okay, so, well, first of all, I would like really to thank you, Eric, to inviting me to give you this seminar. This is really a great opportunity. And as Eric just said, what I'm going to discuss today is phylogenetic challenges in the Retroviridae branch of the tree of life. Essentially, we're going to discuss about virus evolution, virus taxonomy, and the peculiar problems that arise when we investigate the molecular evolution of fast-evolving viruses and retroviruses and RNA viruses in particular, and the spatial challenges that they pose in order to integrate them within, the general tree of life. So let me tell you a little bit what we know right now about viral taxonomy. So this slide is from the virus taxonomy web page. It's the 2009 release. And so far, viruses have been basically organized and classified, I would say. 5 different orders, 22 different families, each one is eventually divided in 39 genera and 476 different species. And there are also, in fact, 65 family, 81 genera, and more than 1,700 species that have not been, in fact, so far assigned to any specific order. So basically, we really know very little right now about viral classification and viral taxonomy. In fact, we know today, some estimates about the number of species that there are on planet Earth. Some estimates say that there are probably about 2 million. Some other estimates say up to 20 million different species on Earth. So if we assume that there are at least as many viral species as bacterial ones, and we probably might have mapped so far no more than 0.1% of less of all viruses. So obviously, whatever we know about the distribution of viruses on this planet today is extremely little, and the picture that we have is extremely partial. This is also due in the inherent bias of our research. Viruses are mostly studied because they are important pathogens. They can be animal pathogens and important plant pathogens as well. So obviously, the research that has been carried over in the past 50 years of molecular evolution of viruses have been really mostly focused, and even before that, on pathogenic species. But definitely, I think it's safe to say that so far we have barely scratched the surface of what viral diversity is on our planet. Now, yes. Okay. Yeah. Can you see me? Okay. Mm-hmm. Maybe it's my microphone. Okay. Okay. So basically, the main message right now that I'd like to give is, in fact, as much as we know about viral evolution and viral classification, we can easily look at the databases, molecular databases right now, and and see that there are hundreds of thousands of viral sequences deposited. There is today we have an entirely database dedicated just for HIV, the HIV Los Alamos databases, that contains probably around 80,000, probably even more sequences of HIV. So there is, in fact, an enormous amount of data out there. And still, in terms of actual mapping viral diversity, we are probably barely scratching the surface. Now, why do we care, actually, about having a general idea of how many viruses and the distribution of viruses on our planet? Because, as I said, viruses are, a lot of viruses and a lot of the viruses that we study actually are associated or can be potentially associated with severe diseases. And especially in the past 30 years, we have seen all around the world and in developing as well as in Western country, the re-emergent or the emergence of a lot of new pathogenic viruses. HIV infection is probably the paradigmatic example. A virus that in fact was unknown until the beginning of the 80s. It was in fact the first human retrovirus, a virus with an RNA genome that is retrotranscribed during the virus life cycle into double-stranded DNA and inserted inside the host cell genome. A virus that was unknown until the beginning of the 80s, and 3 decades later is responsible for one of the fastest growing epidemic in the history of mankind. Same thing for hepatitis C. Right now, there is an estimate that there are almost 280 million people infected with hepatitis C virus. HCV was actually isolated in 1989. So again, and then of course, things like West Nile virus, for example, a virus that historically has been usually localized in Africa, in Eastern and Central Africa, and that in fact just 20 years ago was introduced in the United States, and the next 20 years has spread all over the United States, and now has been has reached also countries like Central American countries, Mexico, Latin America. So definitely has been rapidly spreading also in the American continent. And again, this is not, these are just a few examples. This map is giving you an idea of the major retroviral emerging infections we have been dealing with in the last 30 years. H1N1, for example, the swine flu is the most recent example. So obviously viruses are, have a fundamental interest in terms of healthcare and epidemiology worldwide. They are also very interesting objects to study from the point of view of molecular evolution because especially the retroviruses, and my talk will be today specifically focused on retroviruses, viruses that belong to the so-called Retroviridae family, they have a really peculiar molecular biology. They are double-stranded RNA viruses. They actually have a single, the genome is a single-strand RNA genome, but they are actually diploids. Each virions contain 2 copies of RNA. They have a unique replication cycle, as I said, that is mediated by the RNA-dependent reverse transcriptase, and they can actually, they are pretty ubiquitous in nature. They can be isolated from most vertebrate species. They are associated, of course, with significant human and animal diseases, and they have also limited homology with the retrotransposons that are endogenous retroviruses. And these viruses has actually a very interesting evolutionary feature. They actually replicate extremely fast, especially HIV, and they mutate very fast. The reverse transcriptase is an enzyme that has no error-prone, that has no proofreading activity. For HIV, for example, it's been calculated that there is on average at least 1 mutation per genome per replication cycle. They're very small genomes, usually no more than 10,000 base pairs, but they can accumulate mutations extremely fast. So in a way, from the point of view of the evolutionary biologists, they are extremely interesting objects to study because they allow us to literally see evolution happening in a matter of months or years, and they can be excellent objects to test evolution at the molecular level. So for all these many reasons, actually, they are an interesting object of study. And so let's talk a little bit about the Retroviridae family, which is the main topic of this conversation, and try to figure out, and what I'm going to show you is that in fact retroviruses, in spite of the fact that they have similar genome organization and they can actually behave depending on the family, on the order, the subfamily. They can actually behave in wildly different ways from the point of view of molecular evolution, and it's not really uh, easy, and we will see, not at all clear how we can actually basically study and obtain a general phylogenetic tree related all retroviruses. Now, this classification here, that is again taken by the ICTV website of 2009, the International Committee for Taxonomy in Virology, classify a family called Retroviridae, which are in fact viruses with an RNA genome characterized by the reverse transcriptase and by the cycle of reverse transcription and double-stranded DNA insertion in this host cell genome. And the Retroviridae family is actually divided in two subfamilies, the Orthoretrovirinae and the Spuma Retrovirinae. The Orthoretrovirinae has actually subdivided the subfamily in 7 different genera. And in particular, we will focus on 2 of these different genera, the delta retroviruses. This is the genus that includes different species like PTLV1, PTLV2, PTLV3, and BLV. The PTLV are the primate lymphotropic viruses and are infecting primates in old world monkeys and new world monkeys as well. Monkeys in Africa and in Asia, as well as human. BLV is the bovine leukemia virus. The lentivirus genus is the ones instead that includes HIV1, HIV2, so the immunodeficiency viruses, as well the simian immunodeficiency virus. And finally, the Spumavirinae subfamily so far only has 1 genus, and this includes several species, among which SFB, which is the simian formivirus, BFV, that is the bovine formivirus, and so forth. So what I'm going to do with the next few slides is to talk a little bit about what we know about the evolution and phylogeny of actually delta viruses, and especially PTLVs, what we know about lentivirus, what we know about Spuma virus, and in particular simian formivirus, and show you how essentially these viruses not only evolve with very different biological mechanisms, but also at completely different time scales, and actually integrate all of them in one single phylogenetic tree is not at all a trivial matter. So this is just a classic retroviral genome. Each retrovirus, each member of the Retroviridae family has the same genome. Sorry. And it includes actually a gene for GAG, the capsid proteins, the POL gene for the polymerase and integrase, which include the RT gene, the AMP, which encode for the envelope proteins, and usually there is a region PX that codes for non-structural proteins that often in many of these retroviruses can have regulatory, can work as regulatory proteins. The single-stranded RNA has 2 unique repeats, and identical repeats at the 5' end and at the 3' end. And when the single-stranded RNA is retrotranscribed by the RT in double-stranded DNA, we'll have at the beginning of the end 2 identical LTR, long terminal repeat, with a U3, an R, and a U5 region. I already said that, in fact, the main feature, biologically speaking, of retroviruses is the ability to infect the host cell and retrotranscribe double-stranded DNA, the genome that eventually is inserted by a specific virus-encoded, viral-encoded integrase into the DNA genome, and eventually the virus is expressed, gives rise to new genomes, new variants are assembled, usually in the cytoplasm, and eventually budded out from the cell so that they can infect new cells. This is common to all the families and subfamilies, all the subfamilies and species, genera, and species of retroviruses. However, when we start looking more in detail about their evolutionary history and common history and phylogenetic history, then we encounter several specific problems. Now, this is a general cladogram, really, that kind of depict in phylogenetic terms the taxonomy of some of the major species in the Spumavirinae and Orthovirinae family. This actually phylogenetic tree has been obtained, this genealogy really has been obtained by comparing RT protein. The reverse transcriptase is the only one that has enough actually homology to compare viruses belonging to actually retroviruses belonging to different genera. And as you can see, the one in yellow here are the ones that we will specifically discuss in the next few slides. SFV, which is the simian foamivirus belonging to the Spuma retrovirinae subfamily, the HIV1 and HIV2 and their simian counterpart, the SIV, the immunodeficiency and human immunodeficiency, simian immunodeficiency viruses, and the PTLV, the primary T-lymphotropic viruses that include both human T-cell and simian T-cell lymphotropic viruses, each one of them, of course, belonging to different very different clades in the phylogenetic reconstruction. Now, this tree actually here is actually quite misleading because when we go into the details and we look at the evolutionary history of these viruses, we can immediately realize that actually we're talking, for example, first of all, of a completely and very different temporal time scale. First of all, what is the first problem in trying to obtain a general, phylogenetic classification of these viruses? The first problem is the low homology. Now, these are dot plots, basically plots that compare in a sliding window fashion different genomes at the amino acid level. Here on the left, it has dot plots that comparing an SIV, typical SIV genome with HIV1. The segments on the diagonal represent regions where there is at least 65% or more amino acid similarity in a window of 10 amino acids that is slide along the entire genome. Actually, in this case, along the entire polymerase gene, the reverse transcriptase gene, the one encoding for the reverse transcriptase in these two viral viruses. And as you can see, there are just a few regions that have enough similarity and that can be considered truly homologous. And usually these four major segments along the diagonal are the ones that usually are concatenated together to obtain the tree that you have seen in the previous slide. When you start comparing something like HIV1 and simian foamivirus, still RT gene, so now we're looking at two retroviruses from two different, actually, subfamilies, then you basically see that there is almost no significant similarity. So the moment we even try to, and this is the most conserved gene in these viruses. So already this can tell us that obtaining a tree like this that is truly reliable is not really easy because of the low homology when we start comparing viruses belonging to really different, retroviruses belonging to really different families and subfamilies. The second major problem that we have when we try actually to come up with a general phylogeny of retroviruses is, as I said, the wildly different evolutionary timescale in the phylogenetic tree. We will show in a few slides that actually the origin of simian foamiviruses is dates back probably several million years ago, and probably they share a common ancestor with the bovine feline virus, foamivirus, which is another virus of the same subfamily, as long as 80 million years ago. So this part of the tree, for example, is extremely old. Here in this slide, I put that the putative origin of actually HIV1 and SIV, the common ancestor, is about 2,500 years ago. Unfortunately, I prepared this slide a couple of weeks ago, and just 2 or 3 days ago, I guess, there was a paper published in Science, I believe. I don't know if it's Science or Nature, but there's an important paper from Mike Worobey, who actually has recalibrated in an accurate way the molecular clock to find out the common ancestor, the time of the most recent common ancestor for HIV and SIV. And in fact, he calculated that this ancestor could have existed as long as 130,000 years ago. So clearly, we are talking about at a date that is much older than the one that I put here on my slide, but is definitely much, much younger if compared to the temporal timescale of simian viruses. When we talk about primate lymphotropic viruses, belonging again to a different genus, now we are talking about an evolution which had its most recent common ancestor for all the PTLVs probably around 400,000 years ago. So again, very different time scale, a little bit probably older than the HAV and SAV, definitely much younger than the Foami virus. So clearly, these different, wildly different evolutionary timescale represent a serious problem when we try to obtain a general phylogeny of these viruses and represent them in one single phylogenetic tree and resolve their phylogeny because they have wildly different molecular clocks. And that's actually what this slide is all about. It's a sort of schematic representation of the different evolutionary rates that this virus exhibit. Now, HAV, SAV, and notably, I will talk about this later in more detail, HTLV2 that is actually spreading in injecting drug users, this is what IDUs stands for, usually have evolution rates between 10 at minus 3 and 10 at minus 4 nucleotide substitution per site per year. So HIV and SIV are actually considered among the fastest evolving organisms that we know about on our planet. Now, when we look at primate lymphotropic viruses like PTLV1 and PTLV2, the rate is actually much slower. It's around 10 at minus 6, sometimes even up to 10 at minus 7 nucleotide substitution per site per year. What is interesting here is, again, HTLV2. HTLV2 is the human T-cell lymphotropic virus type 2 that can be actually found in two different populations right now. One is the virus is endemically infecting Native American tribes, especially in South United States and Southern America, and also some isolated pygmy tribes in Africa. But HTLV2 also can be found at a very high seroprevalence in injected drug users, most of which are co-infected with HIV. Now, one thing that is interesting and that we will discuss further is that actually the same virus that is being spreading in two different populations exhibit two wildly different rate of evolution, whether it's infecting drug users and endemic tribes. We will see that this actually has an interesting explanation that has to do with the link of and the correlation between and relationship between evolutionary rate and actually transmission rate. Simian viruses actually exhibit even slower evolutionary rate, around 10 at minus 8, almost similar to the ones of mitochondrial DNA. So as you can see, all of them are retroviruses, RNA genome with reverse transcriptase mediated replication of the genetic information, but wildly different evolutionary rates. And part of our talk will be about investigating why this is so. And of course, this is already makes very interesting this kind of objects. So the evolutionary dynamics of these viruses are also very different, which also in part explains why they have such different evolutionary rates. Simian viruses are usually characterized by low replication rate. They infect a host and almost immediately after infection, they stop replicating within the host and become essentially dormant. And they are essentially characterized throughout their evolutionary history by host virus co-speciation. I show some real studies that demonstrated this point. They seem to co-speciate along with the simian host that they infect. Now, viruses like the simian T lymphotropic viruses, or in general the primate T lymphotropic viruses, instead are characterized by some sort of what we say is a little bit qualitative, but medium replication rate. And mostly they get transmitted by as horizontal gene transfer. Usually, for example, HTLVs, human T-cell lymphotropic viruses, the ones infecting the injecting drug users are transmitted, for example, by blood-to-blood contact through needle sharing between drug users. Sometimes can also be transmitted vertically, like, for example, HTLV in endemic tribes, where the main route of infection is actually from mother to child through breastfeeding. And finally, and both SFV and STLV essentially do not recombine. There are no cases that have been reported of recombination events among STLVs or among simian viruses. HIV and SIV, on the other hand, are characterized by extremely high replication rate. HIV infecting a host can replicate, every 2-3 days in the infected host. By frequent recombination, HIV and SIV are highly recombinogenic, and of course, as we have seen, very fast evolutionary rate. So let me now show you a few specific examples from the literature about what we have learned over the past 10 years on the evolution of these viruses. And let's start from the simian viruses, which are the slowest evolving retroviruses that we know. And this is basically a paper that I've shown a few years back how actually these SFD have been co-speciating with their host. Now, the concept of co-speciation essentially is the concept of a parasite infecting a host. And the idea is that in case of co-speciation, you have a phylogeny that represents, of course, the evolutionary relationships among different host species that are, each one is infected by a parasite. And when we compare the host phylogeny with the parasite phylogeny, in case of co-speciation, one of the scenarios that we can see is that the trees have the same topology. Same topologies, but like in this case, different branch lengths, basically say that every time that during evolution there is a split, a divergence between two different lineages in the host, the same happens to the parasite phylogeny. We can have, this is a form of co-speciation that we can call maybe a topological co-speciation. Now, there is a stronger form of co-speciation where not only the trees share the same topologies, but also share the same time of speciation, which basically means that not only, in fact, the parasite tend to speciate and give rise to new lineages at the when the host gives rise to splits into lineages, but these events pretty much happens almost simultaneously. And finally, we have the strongest way of the strongest possible co-speciation scenario in which the trees of the host and the parasite show both the same topology, the same speciation time, and the same rates of evolution. So literally, host and parasites are evolving in a very similar and correlated way. Now, there are the only example that before the work with simian virus was shown clearly in the literature about co-speciation with the phylogeny of lice and gophers that clearly show some evidence of topological co-speciation with host and parasites seems to have congruent phylogenies. We will see actually that simian viruses not only have a very strong kind of co-speciation, where both the topology, the speciation types, and the rates of evolution in the SFB phylogeny and the simian phylogeny are actually very much the same. So I will skip this for the moment, and I'll give you some of the results. Essentially, the work started as a collaboration with the Center for Disease Control. They have actually sampled a number of simian viruses from a lot of different monkey species, from baboon to mandrill to mangabe to colobine, spider monkeys, and even from a different apes. From each one of these species, or almost each one of them, they also isolated corresponding simian virus. So we had a data set that included mitochondrial DNA sequences. In this specific study, the cytochrome oxidase 2 was used, was sequenced for the mitochondrial DNA. So mitochondrial DNA sequences for